In 1971, the United States government formally declared war on cancer, not as a figure of speech. President Richard Nixon signed the National Cancer Act into law with the explicit goal of doing to cancer what the space program had just done to the moon. Throw enough money, enough scientists, and enough national will at the problem until it simply gave up. The logic felt airtight at the time. Humans had just walked on the moon. Surely a disease was a smaller challenge than that. More than 50 years later, cancer still kills roughly 10 million people every year worldwide. It remains the second leading cause of death on the planet. Treatments have genuinely, meaningfully improved, but the word cure still rarely appears in a conversation about most cancers without several qualifications attached to it. So what actually happened? Was the science wrong? Were the researchers simply not good enough? Was it a funding problem, even though the global cancer treatment industry is now worth hundreds of billions of dollars a year? Or is cancer something fundamentally different from the kind of problem that money and effort alone can solve? It's the last one. And understanding why requires understanding what cancer actually is, which turns out to be one of the most important things most people have never had explained to them clearly. Here's the detail that changes everything once it fully sinks in. Cancer is your own cells, your own DNA, your own biology, running a program your own body wrote, using machinery your own body built, following rules that evolution installed inside you a very long time ago. It isn't a foreign pathogen. It isn't a bacterium or virus invading from outside and attacking your cells from the outside in. And that single fact is the fundamental reason curing cancer is categorically harder than curing an infection. When a bacterium enters your body, it's a foreign object carrying foreign proteins your immune system can learn to recognize and destroy. The line between self and not self is clean. The target is identifiable. Kill the intruder without killing the host, conceptually, is a straightforward strategy. Cancer collapses that entire boundary. The target is you. Specifically, a version of you that has stopped following the rules that keep the rest of you functioning correctly. Destroying cancer without destroying the patient is one of the hardest targeting problems in the history of medicine, precisely because the thing you're trying to eliminate is built from the same materials, running the same biochemistry, protected by the same structures as everything else you're desperately trying to keep alive. This is exactly why chemotherapy, the dominant cancer treatment for decades, works the way it does and fails the way it does. Chemotherapy drugs target rapidly dividing cells, which cancer cells certainly are, but rapidly dividing cells also include hair follicles, the lining of your gut, bone marrow, and immune cells. The drug can't tell the difference. It only recognizes the behavior, rapid division, not the identity behind it. That identity problem sits underneath nearly every difficulty cancer research runs into. Let's go a level deeper into the actual biology, because this is where most popular explanations stop short and where the real complexity lives. Your body contains somewhere around 37 trillion cells. Every single one carries a full copy of your genome, roughly 3 billion base pairs of DNA, the chemical letters that encode your biological instructions. Every time a cell divides, it has to copy all 3 billion of those letters. And copying is never perfect. Each division introduces somewhere between 1 and 10 random mutations, small errors in that genetic code. Multiply that across the trillions of cell divisions happening over your lifetime, and the math alone should concern you. Fortunately, your body has multiple layers of defense built specifically for this problem. Tumor suppressor genes apply the brakes to runaway cell division. Proofreading mechanisms catch and correct copying mistakes before they matter. Apoptosis, a process of programmed cell death, eliminates cells too damaged to safely repair. And your immune system continuously patrols for abnormally behaving cells and clears them out before they can multiply. Cancer only takes hold when enough of these defenses fail at once, in the same cell at the same time. A typical cancerous cell has accumulated hundreds of individual mutations over years, sometimes decades, each one slightly loosening the constraints on normal cell behavior until eventually a cell emerges that grows without limit, ignores signals telling it to die, recruits its own blood supply, and eventually spreads into other tissue. This is cancer functioning as an evolutionary process, and reframing it that way is arguably the single most important conceptual shift in cancer biology over the last 20 years. Cancer cells inside a tumor actually compete with one another. Mutations that help one cell outgrow its neighbors get selected for in exactly the same way beneficial traits get selected for across populations of any organism in nature. A tumor isn't one uniform mass. It's an ecosystem of competing cell populations, each slightly different from the next, each under constant pressure to outcompete the others and survive whatever treatment is thrown at them. This is precisely why cancer develops resistance to treatment. When a drug hits a tumor, it kills the cells most vulnerable to it. But a small number of cells, purely by random mutation, happen to be slightly less vulnerable. Those cells survive, and they reproduce. The tumor that eventually grows back is descended entirely from the survivors, the ones that already carried some degree of resistance before treatment even started. In effect, treatment accidentally runs a selection experiment for drug-resistant cancer, evolution happening in real time inside a living patient during therapy. This consequence can't be fully avoided without solving the tumor's underlying evolutionary dynamics, which is an enormously harder problem than simply finding a drug that kills cancer cells in a lab dish. Now let's talk about why the phrase curing cancer is itself part of the problem. Cancer isn't one disease. It's over 200 genuinely distinct diseases, unified only by the broad characteristic of uncontrolled cell growth, but differing enormously in the cell type they originate from, their specific genetic mutations, their growth patterns, how they spread, how they respond to treatment, and their overall prognosis. Lung cancer, leukemia, breast cancer, glioblastoma, and melanoma are genuinely different problems that happen to share a broad category label. Treating them as variations on a single disease is a little like treating pneumonia, tuberculosis, and asthma as the same illness simply because they all affect the lungs. Even within a single named cancer type, the variation can be enormous. Two patients diagnosed with the same stage of the same cancer might have tumors with completely different genetic profiles, different immune environments surrounding them, and completely different responses to identical treatment. What cures one patient can do almost nothing for another, because at the molecular level, the specific cancer each of them carries is genuinely different biology. This is exactly why clinical trials are so difficult, and why treatments that look extremely promising in the lab so often fail to translate into real patient results. The patient population isn't uniform. The disease isn't uniform. A drug that clears a tumor beautifully in a cell dish might work for 30% of patients with a specific cancer type and do essentially nothing for the other 70%. And the difference between those two groups may not even be obvious until well after the trial concludes. Personalized medicine, the practice of tailoring treatment to each patient's individual tumor genetics, is where the field is heading, and it has already produced real, measurable results in specific cancer types. But sequencing a tumor's genome, identifying which mutations are actually actionable, and designing a treatment around those specific mutations is expensive, slow, and complicated further by the fact that the tumor keeps evolving throughout treatment, changing its own genetic profile in response to whatever pressure the treatment applies. The target is quite literally moving in real time. Now let's talk about the immune system, because this is where some of the most exciting recent progress has happened, and also where the sheer complexity of the problem becomes most visible. Your immune system should, in theory, already be handling cancer. It's meant to be the body's primary defense against tumor development in the first place, constantly detecting abnormal cells and eliminating them before they ever become established tumors. And for the most part, it actually does exactly that. The overwhelming majority of potential cancer cells are destroyed by the immune system long before they ever become a noticeable tumor. The cancers that do become a clinical problem are, by definition, the ones that have already found a way to evade that detection. Cancer cells evade the immune system through a range of mechanisms, many of which involve actively suppressing immune activity in their immediate surroundings. They display surface proteins that essentially signal nearby immune cells not to attack. They recruit immune cells directly into the tumor and reprogram them to suppress anti-tumor activity instead of triggering it. They build a local environment specifically designed to disable the very cells meant to destroy them. Immunotherapy, the strategy of reactivating the immune system's attack against cancer, has produced some of the most dramatic results in recent cancer treatment history. Checkpoint inhibitors, drugs that remove the molecular brakes cancer cells apply to nearby immune cells, have produced long-term remission in patients with melanoma, lung cancer, and other cancers that were previously considered essentially untreatable. Patients who would have died within months are, in some cases, alive years later, but checkpoint inhibitors only work in roughly 20 to 40% of patients with responsive cancer types, and frequently don't work at all in others. The tumors that don't respond have simply found other ways to suppress immune activity that these particular drugs don't address. Some patients also develop serious autoimmune side effects once those immune brakes are removed, because those brakes exist for a real reason, preventing the immune system from attacking your own healthy tissue in the first place. Releasing that restraint to fight cancer also risks releasing it against everything else. There's no clean, universal solution to that trade-off yet. Now let's talk about metastasis, because this is where cancer typically becomes lethal and where treatment becomes dramatically harder. The original tumor, the first site where cancer develops, is rarely what actually kills a patient. Surgery can often remove it. Radiation can often target it directly. The real danger begins when cancer cells break away from that original tumor, enter the bloodstream or lymphatic system, travel to distant organs, and establish entirely new secondary tumors elsewhere in the body. By the time metastasis is detected, it has usually already happened at multiple sites simultaneously. A patient whose cancer has spread to the liver, lungs, and bone at once effectively has 3 separate tumors to treat, each potentially carrying a different genetic profile, each sitting in a different tissue environment, each capable of developing resistance completely independently of the others. To successfully metastasize, a cancer cell has to complete an extraordinary sequence of steps. It has to detach from the original tumor, survive the hostile environment of the bloodstream, evade immune attack while circulating through the body, reach a distant organ, penetrate that organ's tissue, survive in a completely unfamiliar biological environment, and then reactivate growth all over again. Each individual step is technically a potential point where treatment could intervene, but almost none of them are easy to block without also disrupting normal biological processes that rely on very similar mechanisms elsewhere in the body. Then there's dormancy, one of the most frustrating wrinkles in the entire disease. Cancer cells can enter a kind of metabolically quiet state, no longer dividing, invisible to treatments designed to target dividing cells, and nearly invisible to standard imaging. These dormant cells can remain hidden in distant tissue for years, sometimes decades, before reactivating for reasons doctors still don't fully understand. A patient declared cancer-free after successful treatment may still be carrying dormant cancer cells capable of waking back up years later. This is exactly why oncologists typically talk in terms of 5-year survival rates rather than using the word cured outright. Dormancy means being cancer-free today doesn't guarantee being cancer-free a decade from now. Let's talk about the gap between laboratory results and actual patient outcomes, because this is one of the most quietly frustrating patterns in cancer research, and one the public rarely sees clearly explained. A drug works remarkably well in a mouse. Researchers publish the results. Headlines announce a potential breakthrough. The drug then enters human trials, and it fails. This exact cycle has repeated itself hundreds of times across the history of cancer research, and that repetition isn't evidence of incompetence or fraud. It's evidence of a genuine, persistent biological gap between the experimental models researchers use and the actual complexity of human disease. Laboratory mouse tumors are typically genetically uniform, implanted into animals with intentionally weakened immune systems under tightly controlled conditions. Human tumors are genetically diverse, developing inside immunologically complex individuals with completely different genetic backgrounds, different life exposures, and decades of accumulated biological history behind them. A drug that clears a tumor in a mouse is operating on a fundamentally simpler system than the one it will eventually face in an actual human patient. And the very simplification that makes laboratory research possible in the first place also makes its results systematically unreliable as a predictor of what will happen in the clinic. Now, for something genuinely more hopeful, early detection because this is the area of cancer control where the potential for real impact is currently highest and where the science is advancing quickly in ways that don't always make headlines. The single strongest predictor of whether a cancer patient survives is simply how early the disease was caught. Stage 1. Breast cancer has a 5-year survival rate above 90%. Stage 4 breast cancer, the exact same disease biologically, has a 5-year survival rate below 30%. The underlying biology may be identical. The outcome is largely determined by how much time the cancer had to grow and spread before anyone found it. Liquid biopsy, a technique that detects fragments of cancer DNA shed by tumors directly into the bloodstream, represents one of the most promising current approaches to early detection, potentially allowing a routine blood test to catch multiple cancer types before any symptoms ever appear. Several of these tests are currently in clinical development or early real-world use. The central challenge is balancing sensitivity and specificity, catching enough of a genuine cancer signal to find early-stage tumors while avoiding so many false positives that the test becomes useless for screening a general population where most people, thankfully, don't have cancer at all. Large-scale trials of multi-cancer early detection tests are underway right now, and the full results will take years to emerge because proving that a test actually improves outcomes requires following real patients long enough to confirm that catching cancer earlier genuinely translates into more lives saved, not just earlier diagnoses. Finding cancer sooner doesn't cure it in a biological sense, but it dramatically improves the odds that treatments already available today will actually work well enough to matter. There's also an economic layer to this problem that's rarely discussed clearly, and it shapes what research actually gets pursued. Developing a single new cancer drug costs an estimated $2 to $3 billion. From early research through clinical trials to final approval, that kind of investment demands a return, which naturally pushes pharmaceutical companies toward drugs that can be sold at prices justifying that cost. This creates a structural bias favoring treatment over prevention. A treatment can be sold repeatedly to an ongoing market of patients. Prevention is far harder to monetize, since the person who never develops cancer in the first place never even realizes they were the customer. It also pushes development toward drugs that extend survival by clearly measurable amounts, months or years, even when they don't cure the disease outright, because that kind of improvement is easy to demonstrate and easy to justify reimbursing. A treatment that theoretically prevents recurrence in a patient already declared cancer-free is much harder to bring to market, since proving prevention actually worked requires following patients for decades to confirm the recurrence simply never happened. Publicly funded research fills part of this gap, but even that operates on grant cycles and publication pressures that favor incremental, publishable progress over higher-risk work that might not produce results within a typical funding period. The system surrounding the science ends up shaping what gets investigated and how quickly, independent of what the underlying biology actually demands. None of this means the picture is uniformly bleak, and the genuine progress deserves precise credit. Childhood leukemia, once nearly always fatal, is now curable in roughly 90% of cases. Hodgkin lymphoma has gone from a virtual death sentence to a highly treatable disease. Certain breast cancers caught early now have 5-year survival rates above 90%. CAR T-cell therapy, a technique that reprograms a patient's own immune cells to specifically recognize and attack their cancer, has produced remarkable results in certain blood cancers, including complete remissions in patients who had already exhausted every other available treatment. The pattern behind these successes is genuinely informative. The cancers treated most successfully tend to be genetically simpler, more accessible to the immune system or circulating drugs, and built around identifiable genetic drivers that can be specifically targeted. The cancers that remain most stubbornly resistant, including glioblastoma, pancreatic cancer, and most metastatic solid tumors, tend to be genetically complex, immunologically evasive, and physically difficult to reach. The progress is entirely real. The remaining problems are genuinely, deeply biologically hard. Both of those things are simultaneously true. Looking ahead, the next decade of cancer research looks noticeably different from the previous 5 decades, and that difference actually matters. Artificial intelligence is changing how quickly researchers can analyze the massive datasets modern cancer biology now generates. No single human researcher can hold the mutational profiles of 10,000 tumors, the treatment responses of every one of those patients, their varied genetic backgrounds, and the thousands of potential drug interactions relevant to each profile, all in their head at once. Computational systems are already producing genuine insights into cancer subtypes, predicting treatment response, and identifying new drug targets in ways that simply wouldn't have emerged from manual human analysis alone. Messenger RNA technology, which became widely known through COVID vaccines, has cancer applications researchers have quietly been exploring for years. The same underlying platform that can instruct cells to produce a specific viral protein can potentially instruct immune cells to recognize cancer-specific proteins instead, essentially producing a personalized cancer vaccine tailored to each individual patient's exact tumor mutations. Early trials in melanoma and pancreatic cancer have already shown encouraging results. The convergence of better biological understanding, better computational tools, better early detection, and better immunological strategies is producing a genuinely different landscape than the one that existed when Nixon signed that act back in 1971. Progress here has never been a straight line, and it likely never will be, but the direction is real. And that's ultimately the honest answer to the question in the title. We can't cure cancer yet because cancer is one of the hardest problems biology has ever handed to medicine, a moving target built entirely from the body's own machinery, evolving in real time under treatment, spread across 200 genuinely distinct diseases, each with its own biology, each operating through mechanisms tightly intertwined with the normal cellular processes that keep you alive in the first place. Every drug that kills cancer cells risks killing healthy ones alongside them. Every immune activation risks turning against the patient's own body. Every targeted therapy eventually produces resistance. Every success against one cancer type simply reveals the unique complexity waiting in the next one. Cancer will likely never be defeated by a single universal cure. The underlying biology makes that essentially impossible. What's already happening instead is that specific cancers are being understood and controlled one at a time through the specific biology of each individual disease, childhood leukemia first, then Hodgkin lymphoma, then certain breast cancers, with the list slowly growing from there. It will grow slowly by the standards of a news cycle and quickly by the standards of how medicine has historically moved throughout its entire history. The war Nixon declared in 1971 was never really a single war at all. It was 200 separate wars, being fought simultaneously against enemies that keep changing their own tactics mid-battle. Some of those wars are already won. Others are being won right now. Others will take longer than anyone would like. But the people fighting them aren't losing. They're learning. And in medicine, eventually, learning and winning tend to become the same thing.